All right, it's time to get serious. The forest health situation in the United States appears very bleak, just like this photo. A growing number of our most cherished North American forest tree species are under attack by exotic pests and pathogens. This is the most famous case of such a disaster. This is American chestnut. American chestnut after chestnut blight attacked it early in the 20th century. American chestnut was once one of the most valuable trees ecologically and economically in the eastern United States, and especially to the people of southern Appalachia, who used its nuts and its durable timber. In fact, people there called it the cradle-to-grave tree. It was likely that if you were born there, you were rocked in a chestnut cradle, and when you died, you were buried in a chestnut coffin. Well, chestnut blight, which was a fungal disease accidentally imported from Asia, arrived around the end of the 19th century, starting in New York City. And as it moved out of New York City and down through the Appalachians, it killed billions of American chestnut trees, leaving what were called ghost forests. So called because when the trees died, the bark fell off, leaving these white standing stems, and they stood there sometimes for decades because the wood itself was very decay resistant until they finally fell over. Now, this happened a while ago, but the trend is getting worse now. More and more of our species are under attack from exotic pests and pathogens. For example, there's no way for me to name them all, but eastern hemlock, which shaded the streams in our Appalachian forest, cooling them so trout could thrive there, are now disappearing due to hemlock woolly adelgid. White ash, a beautiful landscape tree, and the source of wood for Louisville Slugger baseball bats. Millions of those trees have been destroyed by emerald ash borer, and the list continues. But there are trees in each of these species that survive these attacks. And the reason they survive, at least some of them, as we'll get to, is because in their genes they have the potential to survive. And not only that, what I'm going to tell you today is that these trees have the potential to make an enormous recovery because of the genes that some of these surviving trees carry. On the left of this slide, that is what we call a large surviving American chestnut, LSAs. Some people think chestnuts are gone. They're not. Most of them are quite small, but some of them grow to fair size. Like this tree in Kentucky, it's called the Adair tree. And remember that name, the Adair tree, because I'm going to come back to it. In the center, we have lingering white ash trees in Michigan, so called because all of the other ash trees for miles around have been killed by emerald ash borer, but these trees seem unaffected. And on the right is a member of the eastern hemlock bulletproof stand, a stand in New Jersey. It's not a very big stand, maybe a dozen trees, that seem to be tolerant of hemlock woolly adelgid. These trees may form the basis for repopulation of their species. Of course, we don't really know if they're surviving because of their genetics at this point, or by chance, or some other factor, but we can find out. Here's another thing. If these trees really do have the genetic basis for their resistance, there is a way for them to recover their species. Only it's going to take thousands and thousands of years if they have to do it on their own. Well, I'm here today to tell you that we, and by we I mean scientists and the general public, can greatly accelerate this recovery. And we can do this by what I'm calling a partnership. I've brought here today two props. One is a cell phone, and it's a very old cell phone. And the other is a Petri dish. I'm going to set the Petri dish aside for now and try to remember to come back to it later. Cell phone. What's the cell phone? How does this figure in this recovery? Finding the surviving trees like the ones I just showed you is a big job. It's very difficult to find a large number of surviving trees. Scientists, people interested in restoring these species, we've been trying to find them for years, and we found some to work with, but what we need is a lot more trees, which means we need a much bigger search party to find all these trees. So the first component of the partnership I'm talking about is an engaged public that's out there actively looking for surviving chestnut trees, surviving ash trees, surviving hemlock trees, surviving red bays that are under attack as well. Okay, an engaged public. But in addition to that, we need a way for them to quickly, easily, and accurately report these trees to the researchers who are working on restoring them. That's where the cell phone comes in. So I want the general public to help with this, and you can do this using your cell phone. Fortunately, there is a tool designed just for this application, an app that was developed by scientists at the University of Tennessee and the University of Kentucky. It's called TreeSnap. Using TreeSnap, anyone, even me with a really old cell phone, can, if we find a tree that we think is tolerant or resistant to a disease that is killing off its species, we can use our cell phone and TreeSnap to report it quickly and accurately to researchers working on restoring those trees. We can, by using TreeSnap, provide information on the tree's location, its size, its health status, and send it straight to the people who are looking for these trees to work with. So that's one component of the partnership. Well, who does this information go to? It goes to tree breeders. Now, this person is up in the top of a chestnut tree, and he's got pollen isolation bags on there, and he is doing breeding between chestnut trees. Now, if we find a whole lot of these trees, what the breeders will do is they will breed these trees together to combine their different genes for resistance into the progeny that come from these crosses. It's called pyramiding the resistance genes, and that provides a durable resistance. So that's the second step, tree breeders breeding the surviving trees together. The third step is to clonally propagate the trees. There's lots of ways to do this. It's actually, it's an art to be able to do this, but to clonally propagate trees, you can do things like rooted cuttings or grafting, or what I'm showing here is tissue culture. We can propagate trees in tissue culture, and this is a clone of eastern cottonwood, and it's about maybe 10 trees in there. You can't really see the roots, but these are a clone of eastern cottonwood. And this works with all kinds of trees, as I'll show you later. You might ask, oh, I should say, that once those trees are propagated, then they can go into the field to be tested. Now, why would you want to clone those seeds? Why not just plant the seeds out that come from the crosses and test those? Cloning is a very powerful technology that gives you two very powerful ways to help speed this up. Number one, suppose we just took one tree and stuck it out in the field and tested it to see if it was resistant to emerald ash borer or chestnut blight, and it happens to live. Do we know if that resistance is because of its genetics or by chance? Now take that same tree, clone it 50 times, stick those 50 genetically identical trees out in the field, and test all of them. In fact, test them in different sites and different situations. If they all survive, that is very powerful evidence that the basis for the resistance is genetic. What's the second thing about cloning? Once the very best clones are identified, then we can very rapidly scale these up and make millions of resistant trees and give them to landowners to plant for restoration of the species. Has something like this worked before? Well, kind of. You may know that American elm was once the most popular street and shade tree in the northeastern United States. The streets of towns and cities like Philadelphia, New York, Washington, D.C. were all lined with beautiful American elm trees that shaded their streets. In the 1930s, a fungal disease that was responsible for Dutch elm disease was accidentally imported, followed by a wave of even more virulent form of the disease in the 1960s that essentially wiped out all the American elm trees in all our cities, and they all had to be cut down. It was a very, very sad time. Well, there was an elm that existed at that time, an American elm cultivar known as the Princeton elm. It turned out that by chance, a very popular cultivar, Princeton elm, was already resistant to Dutch elm disease. And since then, other cultivars of American elm have been found that are resistant to Dutch elm disease. And now you see in these same cities and towns, people are once again planting beautiful American elm trees along the streets. These are Princeton elms, beautiful Princeton elms, growing just about 100 yards from here outside the Warnell School on the UGA campus. Well, sounds like it kind of works, but this is not really a good example of what I'm proposing, because essentially these trees were found by chance. I mean, there was already a cultivar, it was already propagated by rooted cuttings, which is how a lot of cultivars are propagated, that already had Dutch elm disease. And I'm talking about something a little bit more complicated. So in order to show you a proof of concept of how the whole plan might work, we're going to look at a current forest health problem that is devastating trees, and this is emerald ash borer, which I just mentioned earlier. And this is still going through our ash populations, killing trees. Actually, it has not reached part of Athens, Georgia yet, but on one side of town, there are already trees infested with emerald ash borer. So you can see the kind of damage it does here. The lower right shows just an entire dead forest of ash trees. So for the proof of concept, what do we need? We need the three components I've just mentioned. First, we need help to find the survivors. Now, we're not going to have Lassie helping us do this, right? But we do have tree snap. And in fact, a number of what appear to be lingering ash trees, surviving ash trees, have already been reported on tree snap. So these trees are available for breeders and propagators to work with. Number two, we need someone to do the breeding. Here's a tree breeder. This is Jennifer Koch. Jennifer is a scientist at the USDA Forest Service Experimental Station in Delaware, Ohio. She has a few lingering ash, green ash trees, but the same thing will work with white ash, that she's breeding together, and she's already produced some progeny of those that she's starting to test. And last summer, we persuaded Jennifer to share some of her lingering ash crossed seeds with us. She sent them to us. And if you haven't guessed by now, what my lab does at UGA is in vitro clonal propagation of forest trees, which is a lot of fun. So Jennifer sent us seeds, and I'll just very quickly walk you through this. The upper left shows the seeds on the trees. They look like little canoe paddles hanging on there. And we start what are called embryogenic tissue cultures that make in culture, this is on a tissue culture medium, in a, you thought I was going to forget about this, didn't you? In a Petri plate. Okay. And what you see there is a cluster, just one of about a dozen clusters on a tissue culture plate making little embryos, each of which is a copy of the original seed embryo. And right there, they kind of look like little baseball bats, which is very appropriate for an ash tissue culture, I think. We pick those off, germinate them. On the lower right, they're germinating still in vitro. Then we can take those out of in vitro conditions, pot them up, and grow them in the greenhouse. And then finally, on the lower left, what you see is a field planting of our very first lingering ash somatic seedlings in a field test about 5 miles from here at Warnell's Whitehall Forest. And we are working with a forest entomologist, Dr. Kamal Gandhi, who is monitoring these. And it is possible that one of those clones may already form the basis for an emerald ash borer resistant cultivar to bring back green ash or white ash. All right, so what I'm proposing here is a partnership. It's a partnership of tree finders, tree breeders, and tree propagators to help restore these trees. Now, each of these components by themselves is not all that novel, except for maybe tree snap, which was just developed very recently. But their combination can essentially, I believe, and used in the right way, can help these trees recover, these species recover, not in thousands of years, but within a human lifetime, which still seems like a long time to us, but it's really a relatively short time for something like this to happen. Now, one other thing I need to tell you is that this approach does not involve any genetic engineering. It doesn't even involve hybrid breeding. It uses the genes the trees already have to accelerate their recovery. At the same time, showing in the early stages that the reason these trees have survived is because of their genes. So it's both proving the concept and making use of it to generate new varieties. Now, I showed you a really depressing picture of American chestnut, right? This is what American chestnut used to look like. And you might ask, can we use this to restore American chestnut, this approach? Well, the truth is that we may have already missed the opportunity to take this approach with American chestnut. Those of you who have studied the history of what happened to American chestnut with chestnut blight know that when chestnut blight first started moving out of New York City, moving down through the Appalachians at the rate of 20 miles per year, killing chestnut trees in its path, some states, like Pennsylvania and Virginia, implemented a quarantine to try and stop the spread of the disease by cutting millions of perfectly healthy American chestnut trees before the blight could get to them. It didn't work. The blight spread the rest of the way through the chestnut's range, but what it did do is eliminate probably a lot of trees that could have carried genes for resistance to chestnut blight, but now we'll never know. But that hasn't stopped us from trying. In the upper right, that is an American chestnut embryogenic culture started from the Adair tree. And in fact, this plate is where that photo came from. This is a culture of, actually it's two large surviving Americans that have been crossed together. One's the Adair tree, and the other is another Kentucky tree called Wayne 3. And at the bottom right, those are American chestnut trees regenerated from that embryogenic culture. This is my plan. And it seems very straightforward, but it's tough getting it going. I'm very excited about the potential for this plan to return American chestnut trees and ash trees and everything else to its former status. I hope that other forest scientists and the general public will also want to become involved and help with this restoration effort. Thank you.